Today's session is humoral immunity, a component of adaptive immunity. In general, immune response is initiated by antigen or an allergen, and the response is created through innate components or adaptive component. Adaptive components are cellular immunity and the humoral immunity, which is mediated through B cells and antibodies, and this is the focus of today's session. The adaptive immune system, which is a specific immune response, and the humoral component of it is basically carried out by antibodies which are produced by plasma cells. If you look at these pictures, you will realize that plasma cells have got a stellate nucleus and a non-staining Golgi, and this is a cartoon that explains or shows the plasma cell, and here is a stained plasma cell. Plasma cells are derived from activated B cells that are produced in the bone marrow. B cells that contains membrane expressing B cell receptors, and these B cell receptors actually are composed of immunoglobulin along with the immunoglobulin alpha and beta chain, and this whole thing is a B cell receptor complex. The T cell support is normally needed for protein antigen by the B cells to create a humoral response. The induction and effector phases of humoral immunity can be depicted or shown with this particular diagram. The first thing is the binding of the antigen to the B cell receptor, and the chemical signaling that either comes through the helper T cell in cases of T-dependent responses, or in other cases with the T-independent responses, and these B cells are differentiated into the plasma cells, which in turn produces antibodies. Normally, a single B cell is capable of producing thousands of plasma cell, and each plasma cell in turn can produce thousands of antibodies in an hour. The effector phase is actually carried out by the antibodies produced by the plasma cells. So we'll go through the details of the induction phase, the binding of the antigen to the B cell receptor, that is actually the recognition phase. And it will be seen that B cells can recognize the antigens as proteins, they can recognize small chemicals or haptens, polysaccharides, lipids, and nucleic acids. Don't forget that B cells are generated and matured in the bone marrow and then released in the circulation as naive B cells that express B cell receptors. If we look at the details of the recognition phase, that is binding of the antigen to the B cell receptors, it's different for the different B cell lymphocyte subsets. There are two lymphocyte subsets, B1 and B2 B cells. The B1 cells, they are mainly produced by the fetal liver hematopoietic stem cells, and they go through different maturation phases, and the immature B cells then express the immunoglobulin on its surface, and finally B1 cells have got IgM and CD5 receptor on its surface. On the contrary, B2 cells, they are produced in the bone marrow by hematopoietic stem cells, and the immature B cells are released into the circulation and they reaches the spleen. And in the spleen you can see that B2 subsets are further divided into follicular B2 cells and marginal zone B cells. The follicular B cells and marginal zone B cell are different as the surface receptors on them are different. The follicular B cells have got IgM and IgD, while the marginal zone B cells have got IgM and complement receptor 2, which is also known as CD21. These subsets of B cells respond to different type of antigens. If we look here, the spleen and other lymphoid organs, they have got follicular B cells and marginal zone B cells. Follicular B cells, if you look closely, have got IgM and IgD expressed on their surface. They respond to protein antigen with the help of T cells, and they both interact inside the lymph nodes and then they differentiate into plasma cells. This T-dependent response has certain characteristic features like isotype switching, high affinity antibody, and long-lived plasma cells. These are the features of the T-dependent responses, and the immunoglobulins that we get at the end are IgG, IgA, and IgE. The marginal zone B cells, they express IgM on their surface, and in the previous slide I showed you they express complement receptor. They recognizes polysaccharides and lipids. Mainly the IgM antibody is produced by the plasma cells, and their features are these plasma cells are short-lived. The mucosal tissue and peritoneal cavity, they have got the other subset, which is the B1 cell, and they expresses IgM and CD5 on their surface. They also recognizes polysaccharides and lipids, and they mainly produces IgM. They also are T-independent responses, and the plasma cells are short-lived. If we want to identify these B cells, different B cell subsets, so it's easy to do it with a set of markers. So follicular B cells, marginal zone B cells, and B1 B cells can be differentiated on the basis of these set of markers. The follicular B cells, they have got CD19, IgM, and IgD are quite high. The marginal zone B cells have got CD19 and IgM. Because they both are B2 subset, and the B1 cells have got CD19, CD5 and IgM on their surface, and IgD is low. Now the second part of this process is the activation phase, where the chemical signals, they help in activating the B cells. The activation and differentiation of B cell take place in lymph nodes. Remember that T cells only recognizes protein in form of peptides. In the activation phase, B cell activation in the T-independent response and T-dependent response is different. The T-independent response, antigens are multivalent. Examples are bacterial polysaccharides or repeating determinants on the surface of viruses, and responses are fast. 1 to 2 days are needed for these responses, and IgM is predominantly produced. This leads to neutral antibody production, and they protect us from encapsulated bacteria. The T-dependent responses are against proteins, and the T cell help is must. Responses are slower, it takes several days, and they produce all IgG isotypes: IgG, immunoglobulin M, A, and E. This leads to the antibodies that are produced. They have got high affinity and the memory cells are also produced. The T-independent B cell activation needs at least two signals, and it happens in two different ways. Now in this particular diagram you can see that the B cells expresses BCR, B cell receptor complex, and this antigen is multivalent. So you can see here's the binding site, here's the binding site, and when this antigen binds simultaneously to two B cell receptors, it will lead to the plasma cell differentiation and immunoglobulin M is produced. But this whole thing requires two signals. The first signal is as a result of the multivalent antigen that simultaneously binds to two binding sites on two different BCRs, and this will cause two signals, one signal through this BCR and the second signal through this BCR due to the multivalent antigen. There's another way of activation of T-independent cells, and that occurs because of the microbe bound to the complement protein C3b or C3d. The B cell receptor produces the first signal and the complement proteins produces the second signal. You can see that C3b, C3d is now inducing the signal through complement receptor 2, which is also known as CD21, which is acting as a co-receptor complex. So in both these situations, there are two signals, signal 1 and 2, that result in activation of the B cell in T-independent responses. The T-dependent B cell activation also needs at least two signals. Here what you see is a naive B cell is bound to this antigen and it is activated, and this is B cell receptor complex BCR bound to the antigen, and it is taken up by these B cells and processed. On the other hand, the naive T cells, they are presented the antigen by antigen presenting cells in the lymph nodes. These antigen presenting cells capture the antigen, process the antigen, and present it with the help of MHC class 2 to the naive T cells. And it needs two signals. The second signal is through co-stimulatory signal CD80-86 on naive T cells and CD28 on naive T cells and CD86 on antigen presenting cell. The activated T cell and antigen primed B cell, they both will then interact with each other. The antigen that is taken up by the B cell, it will be processed and presented with the help of MHC class 2. Now we know that antigen presenting cells are dendritic cells and B cells. The dendritic cells are presenting the antigen to naive T cells to activate them, while the B cells are presenting the antigen to already activated T cell. If we look closely, we'll see that MHC class 2 present the antigen to the activated T cell, and as a result the activated T cell interacts through this co-stimulatory receptor CD40 on B cell and CD40 ligand coming through activated T cell. Now you can see this in detail that the B cell has taken the antigen and it has presented it through the MHC class 2 to the T cell. In the next phase, the helper T cell is activated, CD40 ligand is expressed by the T cell, and it is going to bind to CD40 receptor on B cell. The cytokines are also produced by the T cells to activate the B cells, and this engagement of CD40 ligand and the cytokines are going to cause proliferation of B cells and differentiation of B cell to plasma cells. So this CD40 ligand interaction is very important, and uh, as a result of this interaction there is proliferation of the B cells and cytokine production, germinal center formation, and finally isotype switching, that is, the antibodies switch from one isotype to another. In the T dependent responses, there is T and B cell interaction. The question is, where do the reaction happen? So it depends on the antigen entry route. So how the antigen has entered the body, that will determine where the reaction is going to take place. So if the antigen has entered the blood, it's a blood-borne antigen, it's going to be taken to the spleen, and if it has entered the skin, mucosal lymphoid tissues, some inhaled or injected antigens, they are going to be taken to the draining lymph nodes. Now let's look at this effector phase, that how the B cells are differentiated into plasma cells, and then plasma cells produce different type of antibodies. The interaction of B and T cell in secondary lymphoid organ is actually a very interesting phenomena, but before going into the details we have to refresh our memory about the different parts of the lymph nodes. So these blue structures are actually the B cell zone or the follicles. This area is called as the T cell zone. The inside portion of the B cell zone is the germinal center. The dendritic cells or the antigen presenting cell, they will capture the antigen and then bring them to the lymph node through afferent lymphatics. The lymphocytes, the B and T cells, they will circulate into the lymph nodes through arteries. Now, looking at the initial T and B cell interaction, the dendritic cell will capture the antigen and process it and present it with the help of MHC class II to CD4 helper T cell, and this will happen in the T cell zone. On the other side, in the B cell zone, which is the primary follicle, the antigen, the same antigen which has been presented by the dendritic cell to the T cell, this antigen will bind to the B cell receptor complex, and it will be taken up by these B cells and it will be expressed on MHC class II on these B cells. For this interaction to happen, these B cells, after being primed with the antigen, they have to move from the B cell zone to the T cell zone, and the T cell here has been already activated by the dendritic cells. So now activated T cell and the B cell primed with the antigen come face to face. In the extrafollicular focus, certain other steps will take place which will result in B cell activation. In the extrafollicular focus, these activated T cells, when they are presented the antigen by the primed B cell through CD40 ligand, what will happen? The B cells will be activated and they will differentiate into plasma cells, and these plasma cells are short-lived plasma cell and they will start producing the IgM. So the initial response to certain infections, they are IgM antibodies are produced during that response, and it takes place in the extrafollicular region. But it doesn't last very long, and these T cells which are activated, they are now going to move towards the germinal center, and once they enter the germinal center, they are now called as follicular helper T cells, and they are now in the germinal center, and the B cells that have been activated, they also move to the germinal center. So both the follicular helper T cell and the B cells are now in the germinal center. There's another set of cell in the germinal center, and they are called as follicular dendritic cells. And the follicular dendritic cells, follicular helper T cells, and the activated B cells, they will all result in together the long-lived plasma cells and memory B cells. Let's look at the difference between extra follicular versus follicular steps. So here, as I've already explained, that the T cells, they are going to activate the B cells, which will differentiate into plasma cells, and the plasma cells will be short-lived. The follicular helper T cells move on to the germinal center, the B cells move on to the germinal center, and there are follicular dendritic cell in the germinal center as well. The short-lived plasma cells have a lifespan of 3 days, and they die by apoptosis in secondary lymphoid tissue where they were produced. There is no somatic mutation, and hence the antibodies are low affinity and they are not too much class switched. So you may find IgG, IgM and IgE antibodies. Whereas in the germinal center, the long-lived plasma cells and memory cells are produced, and the plasma cells migrate and live in the bone marrow, and the memory cell release into circulation. So this is what happens in the germinal center. The memory cells that are produced, they go to the blood and they keep circulating in the blood. While the plasma cells that are produced, the long-lived plasma cells, they move to the bone marrow and then they stay there for years. There is somatic mutation and production of high affinity antibodies. The somatic mutation I'll explain in detail, and the high affinity antibody as well, that what are these processes. By the end of this process, isotype switching take place, so the antibodies switch from IgM class to IgG, IgD, IgE or IgA. So as a result of this germinal center process, we get all 5 types of immunoglobulins. Before we go into the details of somatic mutation or affinity maturation, there's something that we need to explain a little bit. There are millions of B cells which are circulating in the blood, and here is just a few that are shown. So if you look at the B cell receptor colors, so it's orange, green, purple or brown, these different receptors, they indicate that they are capable of binding their own specific antigen. And out of those millions of B cells, if just couple of B cells recognizes the antigen, they will be selected, and this selection of a B cell specific for the antigen is called as clonal selection, and these cells are then capable of proliferating and multiplying into huge numbers, and this is called as clonal proliferation, and they then differentiate into plasma cells to produce antibodies, clone of plasma cells, and these are the memory cells that are produced. So out of millions of B cells, just the specific B cell is selected to which will bind to the which will be antigen specific, and then it will make its own army. This illustrates how out of thousands of different naive B cells, only the one with the correct receptor site is selected and cloned. We have already seen that how a naive B cell take up the antigen, process it, and present the antigen to activated T cell in the extrafollicular zone, and then these activated B cells and activated T cell move into the germinal center. In the germinal center, these activated B cells, they proliferate and they make a lot of copies, and when they are making copies, with the help of AID, activation induced cytidine deaminase, they will have point mutation, and their variable regions will have very minor differences. They will all be antigen specific, but because of those point mutation, they tend to have slightly different binding affinities for the antigen. So what will happen now? These immunoglobulin variable regions genes have changed a little bit and their binding strength, the strength with which they bind the antigen, has slightly changed. Now, I showed you in the previous figure there are special cells which are termed as follicular dendritic cells. These follicular dendritic cells are going to present the antigen to these different type of B cells. So these types are all antigen specific, but they have their binding affinities are changed, so they will be presented the antigen. If they bind strongly to the antigen, they will survive. If they don't bind strongly, they will be apoptose, programmed cell death will occur. So in this figure, this particular B cell is presented with the antigen by follicular dendritic T cell, but it doesn't bind well, so it is apoptose. The same happens on this side, but this cell which is in the center, it binds well with the antigen, it gets the survival signal, and it takes up the antigen, and then this antigen is again processed and presented to follicular helper T cell, and this follicular helper T cell will then give its survival signal, and this high affinity B cell is then surviving, and it will be kept for fighting off the infection. So only B cell with high affinity antigen receptors are selected to survive. The process of somatic mutation and affinity maturation provide antibody highest affinity, more efficient to neutralize or eliminate the microbe. Discovering this concept helps in making high affinity monoclonal antibodies and use as therapeutic agent to target a particular disease. We'll talk about this in the next lecture topic. Affinity maturation, we saw that the binding strength of the antibodies is matured. It becomes better and better with the repeated exposure to the antigen, and there is something else that takes place as well, and that is called as isotype class switching. So if we look at the antibodies have got the fragment of antigen binding, the Fab region. It has got the light chains, kappa or lambda. The variable region, the V region, is also termed as CDR, complementarity determining region, and the somatic mutation, affinity maturation actually take place in these regions. So this region is changed or point mutated with the repeated exposure of the same antigen, and the best fit and the strongest fit is found during somatic mutation and affinity maturation. Look at this region. This particular region is called as FC region or fragment crystallizable. This depends on the heavy chain it has. So if it has got the gamma chain, this will be called as IgG immunoglobulin. If it has got alpha chain, then it will be called as IgA immunoglobulin. And if it has got mu chain, that will be called as IgM immunoglobulin. To change one antibody functionally, so the antigen binding site can be same, but if you want to achieve different functions of immunoglobulin, this chain need to be changed from, let's say, IgM to IgG. The constant chain will change from mu chain to gamma chain, and so the function will change as well. So these 5 immunoglobulin have got different chain of the FC region. And this change in the FC region or the constant chain depends on actually the activated follicular helper T cell when it is interacting with the B cell with the help of MHC class II and CD40 ligand. This time they produces the cytokines. Here you can see IL-4, IL-5. These cytokines will determine that which antibody is going to be produced by these plasma cells. So this interaction is very important, and the cytokines are even more important. So here's the examples of what happens with different cytokines. IL-4 will result in different isotypes of immunoglobulins, IgG, and IgE. IL-10 will result in IgG1, IgG3, TGF-beta and IL-5 will result in IgA, and interferon gamma will result in IgG1 and IgG3. So it's the cytokine produced by the activated helper T cell which will determine that plasma cells are going to produce IgG, IgE, IgA or the different isotypes. Now, once these immunoglobulins are produced, these antibodies are going to perform different functions, and just a few of the terms that we remember from our previous lecture are: these antibodies are capable of opsonization, which is coating the pathogen and so that it can be recognized, neutralization of the toxins, bacteria or viruses, the complement activation through classical complement pathway, and the complement proteins then produce MAC formation, or ADCC, which is activation antibody dependent cell mediated cytotoxicity. The effector phases of humoral immunity, the effector function of antibodies, just to list them all one by one. So first of all, the Th2 cells, for example, they will be activated by the B cell antigen presentation, and that will they in turn then produce IL-4, IL-5 or IL-2 to the B cells, and the antibodies are produced. These antibodies can result in the B, and more importantly, the first thing is these B cells will receive the antigen with the help of BCR, then they will present the antigen to the T cells through MHC class 2, and then there will be co-stimulation through CD40 ligand, and the cytokines will be produced. The antibodies then in turn can cause neutralization of microbes and toxins and they can also cause opsonization and phagocytosis of microbes. So the antibodies bind with the pathogen and they will be recognized by phagocytes like monocytes, macrophages and neutrophils. Sometimes the cells are bound to the antibodies, and these antibodies are recognized by natural killer cell, and these are toxic cells. They can then cause apoptosis of the cells. So it is antibody dependent cell mediated cytotoxicity. The cell is killing the other cells. And lysis of the microbes is a result of complement activation. So we know the classical pathway, alternate pathway, mannose binding pathway. Out of these three, the classical pathway is antibody dependent, and this antibody dependent pathway will result in MAC formation. That MAC will result in lysis of the microbe. At same time, they will also result in the other complement proteins like C3b, which will bind to the pathogen, and they will be recognized by complement receptors, and these complement will then engulf the pathogen. These macrophages will then engulf the pathogens. And finally, they are also capable of inducing neutrophils to the site of inflammation, to the site of infection, and the inflammatory response is induced or enhanced. As a whole, now if we have to summarize all the features of the humoral immunity, we can see that the antigen bind to IgM and IgD in the form of B cell receptor on naive B cell, and the B cells are activated. They have clonal expansion and proliferation. They differentiate into different type of cells: the memory cells, the long-lived plasma cells, during which they go through isotype switching, affinity maturation that result in different immunoglobulin formations, and this process is very much dependent on CD40 and CD40 ligand interaction, and the cytokines that are produced determines which type of immunoglobulins will be produced, and the somatic maturation or rearrangement of immunoglobulin genes will tell you that how strong or how perfect is going to be the binding between the antigen and the receptors. And the plasma cells then move from the germinal center to the blood or the peripheral lymphoid tissues, and the memory cells, they move on to the bone marrow where they live for many years. Thank you very much.